the important role in the plant efficiency against the pathogenic fungal organisms. Transgenic plants expressing the genes of the chitinases and the gluconases. Plant respond to attack by the pathogenic microorganism by the induction of expression of a large number of genes encoding for the pathogen-related proteins, many of which are the believed to have the role of deficiency. The major classes of the PR proteins are the tobacco, PR1, PR2, PR1, PR2 is the beta-1,3 galactogluconesis. The PR3 is the chitinases. PR4 is the heaven. PR5 is the something like the osmotin. The ability of the hydrolytic enzyme to break chitin and the glucan, the cell walls of the fungal pathogen, has been exploited to develop crops resistant to the pathogens. Fungal pathogens and it can be hydrolyzed the by the enzyme chitinase. Chitin is the linear homopolymer of the P14 linked ester glucose. Is this. On the other hand, of the A13 gluconase is known the degrade glucans, which are also present in the fungal cell walls. The synthesis of the chitinase and the glucans is known the occur as a response to pathogen infection. When both enzymes are the simultaneously present, the fungal growth is more effectively inhibited inductions of the chitinase is often by coordinated with the induction of the specific P13 gluconesis and other pathogens of the BPR. The types of the chitinases most extensively studied in the plants and endochetinases, which is the randomly hydrolyzed internal P14 linkages of the chitin, releasing oligosaccharides of the gluconate. Some of class 1 chitinases are the localized in the vacuoles, whereas the other chitinases are the located coplastically. Therefore, the protect plants in the areas such as the fungi, antifungal compounds need to be present in the intracellular spaces rather than the inside the cell. The prospects for the genetically engineering plants with improved the resistance by the transformation with the chitinase genes seem encouraging, and the approach is known being the utilized production of transgenic plants around the world. The transgenic tobacco and the rare plants, rare plants of the containing gene with the constrict promoters have been shown to exhibit the higher basal levels of the chitinase and contaminate the increased resistance of the Rhizoctonia solani. In another example, tobacco plants are transformed with the basic chitinase from the tobacco under regulation constitute the promoter accumulated up to 120-fold more active chitinase than non-transformed plants. The transformed plants were found to be no more resistant to the fungus Cercospora nicotiana than the controls. It denotes that the chitinase genes, perhaps in combination with the other genes, including the antifungal products, will provide a means for the improving resistance against some pathogens. An alternative approach tightens either from the microbial or plant sources used the means of biological control agent. This approach is supported by the results obtained the injected bacterial chitinase, which inhibited the development of powdery milk, polypeptides. The many bacterial chitinase gene has been the inserted from the cell bacterium and the integrated in it tobacco. The chitinase gene bean from the bean Phaseolus vulgaris has been expressed to the tobacco and brassica. The combined expression of chitinase and the glucanase is the transgenic carrot tomato tobacco resulted in a much more effective prevention of the fungal disease development. Transgenic plants with the improved the resistance against the insects. The insects and the pests that contribute the significant protection crop yield around the world. Insects not only cause the direct loss due to the agriculture produce, but also indirectly due to the role as vectors of the various plant pathogens. Most of the pests attack the plants in the flowering and the fruiting stage, causing the huge yield loss to the farmers because a large amount of the money is spent on the raising the crops. The damaging to crops is mainly caused by the insect larvae and some of the extent this chemical pesticides can be used to some extent, but the most pesticides spray has washed away from the plant surface and it also accumulates in the soil, causing environmental pollution. Sometimes it is difficult to deliver the pesticides to vulnerable parts of the plants, such as the roots, stems, and fruits. Most of the synthetic pesticides are the toxic or non-toxic organisms, particularly humans and the animals. Hence, the alternatives to the chemical pesticides have to be developed in plants, providing the resistance to insects. With the intervention of the genetic engineering technology, many transgenic or genetically modified GM plants were developed with inclusion of insect resistance that the well-known insect resistance approach is the reduction of the bacterial gene bacterial bacterial gene the plant gene expresses the insect cell the proteins which is the S in the plants. The other genes which are the used in the insect control, including the coffee, trypsin inhibitor, alpha amylase inhibitor, and assuming the snowdrop lectin proteinase inhibitor. BT plants are the produced by the transfer of the gene from the Bacillus thuringiensis, coding the for a the gene expresses the insectile proteins which kills the pest cropland. The other genes which are the used the insect control, including the coffee trypsin inhibitor, alpha amylase inhibitor, and snowdrop lectin, the protease inhibitor, etc. Bt plants are produced by the transfer gene from the Bacillus thuringiensis, coding for a histidine protein. Protease inhibitors, proteases inhibitors are the able to protect plants against the insect attacks by the interfering with the proteolytic activity of insects in the digestive gut. Lectins are the carbohydrate binding the proteins found in many plants are the toxic to the insects, especially against the sap sucking insects. Alpha amylase inhibitors, these inhibitors the alpha amylase with the are the postal importance of the digestive enzymes of insects. BT plants are the transgenic plants produced with the transfer genes from the Bacillus thuringiensis to the crop plants, which provide the resistance to the pests. The genes coding for the cry proteins have been isolated from the bacteria and transferred into the plants through the various gene transfer methods. Many of the agriculture important crop plants have been the transferred with the Bacillus thuringiensis genes. The BT is the short form from the soil bacteria. Bacillus thuringiensis, which is the synthesize the crystalline proteins, is called next one. BT plants are the produced by the transfer from the gene Bacillus coding the for the crystal protein. Protease inhibitors, plant protease inhibitors are the able to protect the plants against the insect attacks by the interfering with the proteolytic activity of insects. That is that. Lectins are the carbohydrate binding proteins found in many plants and the toxic to the insects, especially against the sap-sucking insects. Alpha amylase inhibitors. These inhibitors are the alpha amylase, which are the most important digestive enzymes in insects. BT plants. BT plants are the transgenic plants produced with the transfer of genes from the Bacillus thuringiensis to the crop plants, which provide resistance to the pests. The genes coding for the cry proteins have been isolated from the bacteria and transferred into the plants through the various gene transfer methods. Many of the agriculture important crop plants have been the transferred the bacillus thuringiensis genes. The bid is the short form of the soil bacterium. Bacillus thuringiensis, which is inside crystalline the proteins called endotoxin. The Bacillus thuringiensis bacteria codes for the crystalline protein, which are the toxic to insects but non-toxic to human and animals. Currently, BT is the major transgenic crops growing the worldwide and occupies the first position in the distribution. Several transgenic plants, residents, insects have been the produced. For example, tobacco, cotton, tomato, mustard, corn, brinjal, etc. Bacillus thuringiensis bacteria is the first discovered in 1911. The Japanese biologist Shintine Ishwatari, that is the gram-negative bacteria and soil bacteria, produce toxins which are harmful to various insects like larvae of moths, butterflies, beetles, cotton, bollworms. Bacillus thuringiensis forms the proteinases, then insecticide the endotoxin, also called protein, the cry protein. which are the encode the cryogenes in the most strains of the bacillus thuringiensis. The cryogenes are the located in the plasmids. Beta-toxin genes. Phytoxins have the specific activities against the species of the order Lepidoptera, that butterflies, Diphtheria, flies and the mosquitoes, Coleoptera, beetles, Hymenoptera, bats and the ants and the flies, and nematodes. The Bacillus thuringiensis as an important reservoir of toxins for the production of the biological insecticides and insect-resistant genetically modified crops. Mode of action of beta-toxin. The gene coding for the beta-toxin has been the inserted into the crop plant. And transgenic plants resistant to insects have been the used. These BT crystal proteins are the expressed in various parts of the transgenic. The BT plants, when the insects attack and eat the different parts of the transgenic plant, the cryotoxins attached to the insects. These proteins are the cleaved by the digestive protease of the insect. Then the prototoxin gets activated within the gut by combination of the albumin pH 7.2 to 8.5 and proteolactic enzyme. The activity form of toxin protein gets itself inserted into the membrane of the gut epithelial cells of the insect. This results in the formation of ion channels through the which are there the occurs and the excessive loss of cellular ATP. As a consequence of the cellular metabolism cases, this insect stops feeding the becomes dehydrated and finally dies. BT transgenic crops showed the effective protection against the insects damage. Many genetically modified plants were given approval by the USA in the mid 90s. Some biotechnological companies with their own trade names introduced the several transgenic crops into the fields among all cotton and maize BT crops, met with the huge success and the growth in millions of the hectares worldwide. BT cotton is a transgenic cotton crop produced by the transforming the insectile resistant cryogens from the soil bacteria. Bacillus thuringiensis cotton, it is the one of the major fibers crops grown in the more than the 80 countries around the world. Cotton is the prone to attack by many types of the insect pests and various stages of the development. The major sucking pests of the cotton is the aphids, seed strips, and whitefly. The bollworms are the most important. The major sucking pests of the cotton are aphids, seeds, the thrips. The bollworms are the most important tissues, the feeders, and the highly damaging. Three types of the bollworms: American bollworm, pink bollworm, and spotted bollworm, and refer to the bollworm complex are the most damaging pests of the cotton. Amongst them, helio, Heliophoramum as the key pest in the causing 80% loss of cotton. The first BT cotton plant was developed in 1987 in USA by the Man Center. Delta and Pine companies later on the research work on the development of transgenic was intensified all over the globe and several transgenic plants were developed. India cotton is being the cultivated large area of the major sources of employment, textile industry. In India, BT cotton is the first GM crop approved in 2002, now largest producer of the GM cotton. And BT brinjal, PT brinjal is the genetically modified crop created with the inserting the cryogene from the soil bacteria Bacillus thuringiensis into the brinjal similar to the BT cotton. The major insects are to brinjal are the shoot borer and the fruit borer. In India, BT brinjal was always approved for the permission of the field trials in 2009. Since the brinjal was food crop, there is a lot of opposition from the public and regarding the safety. The matter reached the Supreme Court and the expert of the committee appointed the Supreme Court and recommended the 10-year moratorium field trials of the brinjals, the called the genetically modified crops in the India. What are the advantages of the transgenic plants? The biotic stress. Transgenic plants are the produce the modifying its the genetic material to produce the desirable traits to the characters. Various genes coding for the specific traits are the introduced to the plant's genetic makeup using a set of several biotechnology techniques, collectively known as the recombinant DNA technology. The inserted genes can be transferred from the same or different species of completely unrelated source like the bacteria or the animals. Once transgenic plant is created, the newly introduced gene is injected in plant along with the rest of the its genes. Transgenic plants produce the high levels of the resistance against the various biotic stress and improvement of other traits and importance. Transgene is the expressed in all parts of the plants, including the roots, leaves, and the interior regions of the stems and the fruits. Transgenic plants can be produced against the various diseases and pests, hence the use of the chemical insecticides for the control of the insect pests reduce the drastically. The transgenic crops is the various antibacterial and toxin proteins are the produced within the plants and not exposed to the outside, and these are the environmental friendly. Since few rounds of the chemical pesticides are the sprayed, in case of the transgenic plants, it will minimize the soil contamination and the pesticides runoff. It also reduce the pesticide spray and risk the health hazards associated with the use of the insecticide also reduces. The transgene technology as the results in improvement of yield levels is the directly improves the margin to profit to the farmers. Next one, integrated pest management. It is the main different definition agreed by the Universal Food and Agriculture Organization. NGOs such as the Pesticide Action Network and the pesticide industry. The effective and environmentally sensitive approach of the pest management that relies on combination of various practices. The term integrated pest management was the firstly used the Smith and Van Dorn Bosque in 1967. IPM is the holistic and eco-friendly approach to sustainable agriculture that the focus on the managing insects, weeds, and diseases through the combination of the cultural and physical, biological, and the chemical methods that are the cost-effective, environmentally sound, and the socially acceptable. This includes the responsible use the crop protection and plant biotech produces integrated pest management, which is the aims keeping the pest population at below the economic threshold levels by the applying all the available alternate pest control methods. Next one, IPM is the decision. Principles of integrated pest management. IPM is the decision-making process that is help prevent pest problems. With the IPM programs, all the information and treatment methods are the considered in order to manage pests. These elements should be effective, affordably, and the safe for the environment. The, the prevention is the kept step to the in IPM. Culture and the physical controls can be the constant preventive. The prevention and the supervision of the of harmful organisms should be the achieved or supported among their other options. Crop rotation, use of the adequate cultivation techniques, use of the resistant tolerant cultivators and standard certified seed and the plant planting material, is the balanced fertilization, liming and irrigation, preventing the spreading of harmful organisms by the hygiene measures, the production and the improvement of the important beneficial organisms by the adequate plant protection measures are the utilization of the ecological infrastructure inside and outside of the protection. Identification, not all the plant damage or decline is caused by the pests. So pests and beneficial organisms are the identified. Certain abiotic factors also cause the damage to the plant. Many types of the pest damage look the same, but the control practices for the individual pests very. So it is very important to know what the problem is before choosing the treatment. And the pest identified the information on its the biology and life cycle is to the known. One has to recognize the beneficial organisms before the dedicating the treatment are required. For example, to if enough the lady beetles are the present in the aphid colonies, the pesticides may not be needed the control the aphid. Monitoring is given information need to the make right decision the managing pests. Regular monitoring makes it is the possible to tell where the pests are the size of the population and if it is the increasing and decreasing the monitoring done by the visual observation and the counting measuring the methods. This helps the when declaring the whether treatments are the needed and the most effective time and place based on the results of the monitoring. The professional user has to the decide the whether and the when the to apply plant protection measures. Accurate the threshold values are the essential components for the decision making for harmful organism threshold levels defined for the region specific areas. Crops and the particular climate condition must be taken into the account the before treatment, where the feasible pest and the beneficial organisms, the population are observed as well as the pest damage and the environment. Harmful organisms must be monitored by the adequate methods and tools. The observation in the field as well as the scientifically proper weighting, forecasting, and the early diagnosis systems where the feasible as well as the use of the advice from the professionally qualified advisor and including the methods and tools, treatments and available information is the IPM integrated pest management program to select the best treatment are used including the cultural, biological, physical, and mechanical and behavioral chemical methods combination. The goal is the control pest with a little impact on the environment, sustainable, biological, physical, and other non-chemical methods, and prefer to the chemical methods for the provide the satisfactory pest evolution. The effectiveness of the pest management plans are the considered the based on the records on the use of the cultural, physical, mechanical, chemical, and the biological methods. And on the monitoring of harmful organisms, the professional users should be check the success of the applied plant protection measures. Components of the integrated disease management. The integrated disease management involves the need based on the uh, pesticides only the when the disease incidence reaches the economic threshold. Integrated disease management involves the four components. post resistancy, biological control, cultural control, and chemical control. Integrated insect management (IIM). The management of insect pests rarely release the single control method. Usually, variety of the practices and the integrated to maintain the pests and acceptable levels of the minimize the chance that insects will be adapted to the any one of the management practices. The aim is not to eliminate all pests, but the rather is to produce the pest populations to tolerate levels. Some pests are the tolerably and the essential, so that they are the natural enemies remain in the crop. The management practices use the against insect pests, including the pest resistant, to tolerate the varieties and the cultural, physical, mechanical, biological, and chemical controls. Cultural practices. The cultural practices are the integral part of the IPM, timely plowing, the selection, healthy seeds, timely planting, rising healthily, nursery, removal of the weed from the field, balance on the fertilizers, and physical, chemical, mechanical practices, remove and destruction pests infested the plant pots, use of the physical barriers such as the row covers of the trenches prevents the insects from the reaching of the crop. For example, row covers can help the prevent early season damaging the cucurbits and by the cucumber beetles and the plastic lined biological control pests. Chemical control measures and resistant crops is the pest resistant plant varieties developed by the classical plant breeding recently. The use of the genetically engineering pest resistance in IMS has the proved the effective, such as the BT corn, potatoes. There are also special uses of the genetic techniques on pests themselves, such as the sterile male insect releases. Integrated weed management. Integrated weed management is defined as the combined use of all the methods of the weed control, cultural and physical, chemical, and the biological methods, such as the balanced way of use. Application of IPM. Advantage IPM, slower development of resistance to the pesticides, reduce to the health risk of the spray operators, reduce the chemical contamination of food and the environment, reduce the dependency of the on chemicals, and IPM is the step towards the sustainable agriculture. Better plant health, giving the improved quality and production, reduce the crop loss, reduce the chemical application will reduce the risk to the health of staff members. Decreased use of the chemical application will reduce this discrimination and the disfigurement of the holding. Decreased use of the chemical application may result of the financial saving. Next one, the environmental improvements made of the facility, the implement of the integrated pest management program will enhance the long-term stability of the holding over and the above protection against the pests. IPM may the only the solution to the some long-term pests and the problems where the chemical application has not worked. Integrated pest management ultimately allows the institution of to have greater control over the knowledge and of pest activity in their facility. Integrated pest management is the pest management technique of choice for the major institutions. Disadvantage to the integrated pest management. IPM is the requires more staff time than the traditional pest management. Even the implementation of the contracted the pest management company, IPM requires the coordinate effort all the staff members for the proper implementation. Integrated pest management initially be more expensive than traditional pest management. Integrated pest management in India mainly the they can introduce in India 1974 to 1975 in cotton and rice. The integrated pest management is a farm in the 12th year plan of the EFC National Submission of Plant Production and the Plant Quarantine was introduced from the year 2014 to 15. Strengthening and modernization of the pest management approach in India has become one of the components of this submission with the mandate to popularize adaptation integrated pest management through the training and demonstration crops in the interior promotion of the biological control approaches in the crop production technology. Thank you.